And good afternoon, molecular genetics friends. So, last week, when we last discussed, we were talking about the role of dideoxynucleotides in sequencing and basically going through the basics of the timeline of sequencing. How initial DNA sequencing using Maxim Gilbert style technologies was using rather caustic and nasty chemicals to cleave DNA at specific nucleotide sites, and you would do a partial cleavage, and it was based on the fact that you had an end label, and then you would run this out on a gel and you'd get this ladder and you could read the nucleotides. The real breakthrough in sequencing came with the use of dideoxynucleotides, or chain terminators. Again, you have a dilute solution of dideoxynucleotides in the presence of also the deoxys, and so you end up generating stops at every single nucleotide around a large genome, or a large piece of DNA. My phone is dinging here. I'm going to go shut this thing off. All right, so we got dideoxy sequencing, and this is really a breakthrough because it now allows for the automation of DNA sequencing, in which we can use fluorescent tag nucleotides that are labeled with the dideoxy, and every time you stop, you are putting in a specific nucleotide, and it has a very specific fluorescence wavelength. And so by running the samples out not on a gel, but on a capillary electrophoresis system, and you don't have to run one lane for A, one lane for G, one lane for T, one lane for C. You put it all in one tube, and then you have a laser scanner at the bottom reading the fluorescence as it comes out, and you get what we call an electrophoreноgram, these peaks of nucleotides, and you have what's known as a base call, in which the system is able to actually tell you what the nucleotide is. And so it is reasonably expected that you could probably get 800 nucleotides in a run, and with standard gel electrophoresis dideoxynucleotides, you might be able to get 300. With Maxim and Gilbert, you're about 100 or 150. So the technology is dramatically increasing in terms of the reads. And in the mid-'80s, the idea of doing the Human Genome Project comes about. In that, one guy named Lee Hood, another guy named Eric Lander, they were developing some of the technologies that would essentially automate this system. And initially, it was thought that the way to do this was to first generate an ordered set of clones around across the chromosome. So let's just take, for example, the X chromosome, which is going to be one of the first chromosomes to actually be sequenced. I'm going to shift markers here. I don't like this one. And these big markers are fun, but they do die rather quickly. So, all right. So we're going to start with the X chromosome. And hold on a second. If I could pause, I would, but my machine doesn't let me pause. So the notion was you would separate out X chromosomes from a sample, and then using genomic library techniques, basically chromosome walking techniques, you'd establish an ordered array, well, let's go ahead and make an overlap, an ordered array of clones that each one was maybe 50,000 nucleotides. Around this time, the yeast artificial chromosomes had the ability to hold upwards of maybe even a million nucleotides. And so you develop an ordered set of clones, and then that was going to take 5 years. And at that point, you then begin sequencing them. And you're just going to start sequencing, starting from clone 1 and working your way down. And it was going to be a slow and tedious process, required a lot of work that really never went anything more than just developing the ordered clones. And so the concern was, we're going to have all these postdocs doing this work, and where are they going to get out of it? Because until you get the sequence data, there really isn't much there. You just have established this ordered set of clones. So as time goes by, the sequencing technology begins to also increase and gets cheaper, and the ability to do essentially robotic sequencing comes about. And what you can do here is have a 96 well plate, 96 samples, and you have a system that can run the sequencing reaction in all 96 wells, and then begin running the samples out in capillary electrophoresis, and you could get something like 50,000 nucleotides or so out of one plate. I mean, you could really start to actually sequence these things. And so these sequencing bots were rather elaborate machines that could do this, required actually massive amounts of cooling because they generate a lot of heat, and so the air conditioning systems for these genome centers was quite impressive. So this brings about then an idea that a researcher, scientist, bit of a rebel, as they sometimes are, a guy named Craig Ventner, developed a company called Celera, and he invested in 300 of these sequencing bots. And he began doing a little bit of calculation that rather than developing an ordered array, if you simply took a genome, say, and you started out with the Drosophila genome that has got around 1 million nucleotides, and let's see, what is it? Anyway, the calculation being that if you say, just as an example, you have a genome with 1 million nucleotides, if you sequence just randomly 3 million nucleotides, you just sequence until you sequence 3 million nucleotides, you will statistically cover the entire genome. And then you have the task of taking all this data, putting it into a computer that will then look for the overlaps and begin building what they call contigs, or these contiguous sequences. And so they did this with the Drosophila genome. And so in it was probably like around 95 or so, the Drosophila genome was published. And it was kind of an amazing thing, is that everybody at the fly meeting got a disc with the entire fly genome on it. And this was a trial run. It took them about 3 weeks to do this. And at that time they had what was supposed to be the largest parallel processing microcomputer array outside of the Department of Defense to actually take this data and develop the contigs. The next step was the Human Genome Project, and it really only took them about 3 months worth of sequencing. So the idea was you just develop a whole series of just random DNA fragment clones. And so you've got a plasmid, and so then you have this unknown piece. The plasmid provides a known sequence for the primer, and you then begin running your way through. And they had robotic systems that would just pick individual colonies, put them into a plate, isolate the plasmid, run the entire sequencing thing completely automated, and each bot could run something like 300 plates in a day. And with that much sequencing capacity, you don't have to bother with the very careful ordered array. You just massively sequence it. Now, Ventner was a bit of a difficult man to deal with, and the head of the Human Genome Project is a phenomenally thoughtful man named Francis Collins, and he was pretty much the exact antithesis of Craig Ventner. He was a very thoughtful, very stepwise thinking scientist, and Ventner is a pretty serious showman. They decided, though, that really Celera was going to actually, their way was in fact the way forward, and the Human Genome Project agreed to cooperate with Celera, and they began sequencing the human genome in this way. So they get the data, they then begin putting together the contigs, and there's still actually little gaps of sequence that aren't known, and you can't link up all the contigs in one large, gigantic sequence, but it's pretty darn close. And they were able to, in fact, sequence the human genome, and the idea was, in fact, they put this out into the National Center for Biotechnology Information Database. And so all of a sudden, within literally a few weeks, BLAST became huge with all the human genome data. And the next step is that they gave away the data for free, but really for pharmaceutical companies and people that needed to have better information about the genome, there was what was called the annotated genome, in which they would then take this sequence and, using a variety of computer algorithms, try to identify all the different genes. And so the genes that had already been cloned, it was just a matter of matching the sequence up. Other things they still do is use essentially artificial intelligence to look for regions that look like they could be genes. One thing we know is that genes tend to have a much greater GC content. And you identify the genes, you then translate the DNA, look for open reading frames, and you begin developing these known amino acid sequences, these known proteins. And so even to this day, there are proteins that have been identified, genes that have been identified in the Human Genome Project that we don't really know much about what the proteins do. We know that the possibility exists for them to be coded. There's a lot of information still left to be figured out now, even 20 years later, but it is an amazing tool, and the sequencing capacity of these different centers is really pretty impressive. So this process of random sequencing and essentially doing a 3x coverage as a way of sequencing the entire genome, that becomes really the standard. And so now pretty much every organism of agronomic importance, every model organism, lots of different organisms have their genomes sequenced. And so what do we know from all of this genome sequencing? There are a lot of things that are actually interesting to think about. So I'm going to jump into Chapter 6, which is an introduction to gene sequences and gene numbers. And it's an interesting story to think about. And we've talked a little bit about this in terms of genome number and thinking about the N paradox and what is the minimum number of genes required to be different kinds of organisms. So a couple of things that come out when you begin sequencing genomes is that there's a lot of DNA in complex eukaryotes that doesn't code for genes, that is repetitive DNA. And so this is going to become an interesting kind of question that we're going to be thinking about. So if we look at an intracellular parasitic bacterium, like a rickettsia organism, as they're called, these are bacteria that are not free living. They actually require a cell to live in. They've got about 500 genes. A standard bacterium has about 1,500 genes, 1,500 genes. Unicellular eukaryotes, about 5,000 genes. You get to multicellular eukaryotes like a nematode, about 13,000 genes. Plants require a bit more in terms of being able to carry out photosynthesis and all the different kinds of metabolic things they have to do. They're around 25,000 genes is the minimum number of genes that found in higher plants. And then in mammals, what we think of as the top of the heap, we're really only at about 25,000 genes. And so as a Drosophila investigator, it was a little disconcerting to think that the organism that I was grinding up had 15,000 genes and I only had 10,000 genes more. Prior to the Genome Project, prior to 97 or so, it was estimated that the human genome had around 100,000 genes. And so I was within almost an order of magnitude more complicated than the organism I was grinding up. Well, it turns out that no, we don't really have that many more genes. Now, just as an aside, we'll get to it in a bit, the transcriptome of advanced mammals and organisms with nervous systems and vertebrates in general ends up being way higher than the transcriptome of insects or lower eukaryotes. this owes to the increased amount of differential splicing. That's why the number of introns within the genes is such a critical point, because differential splicing allows you to generate a whole great number of different gene products from a set of genes. So while we may share with almost identical and not that many more genes between flies and humans, our ability to carry out complex splicing increases the complexity of the number of RNAs that we can generate. So the prokaryotic gene numbers, they range over an order of magnitude. So we've got the minimum number is about 500, and then in most bacteria, it's about 1,500. So the genome number is proportional to the genome size in prokaryotes, and this makes sense because prokaryotes tend to be very complex or very compact genomes. They don't have a lot of repetitive DNA. We talked about this when we were looking at Koch curves. If you do a Koch curve on a bacterium, they will exhibit a single inflection of DNA hybridization, whereas with very complex eukaryotes, we have these multiple levels of inflection, which represent these multiple numbers of either highly repetitive sequences in the genome, moderately repetitive, or single copy messenger RNA. So the prokaryotic gene numbers, they range over a pretty wide amount. There are DNA segments that are present in pathogenic bacteria that are absent in non-pathogenic bacteria. There is DNA that can be transferred from one cell to another, what's known as horizontal gene transfer, and that is a way of transmitting different kinds of genes. This is how antibiotic resistance is spread. So now, as we were talking about, although this may have been something we were talking about in evolution, the number of genes versus the genome size begins to diverge once we get into complex organisms. And this is a question. How is it that organisms that have essentially the same number of genes have dramatically different genome sizes? Or another way of phrasing this is, we would think that the more complex an organism is, the bigger their genome is. But it doesn't hold that way. If we look here, we've got yeast down here, here's flies, here plants, Arabidopsis, little mustard. Rice actually has got a pretty large genome, mice and humans. And so it works fairly well if we limit the number of organisms that we're looking at. But it doesn't really track until you actually take the minimum number of genes required in each different taxa and then plot that with complexity. And that requires, say, looking at all mammals and identifying what mammal has the minimum number of genes, and then comparing that to a plant and identifying which plant has the minimum number of genes to make it actually a plant. And then you actually get a pretty good correlation. So the total gene number is known for lots of different eukaryotes. And we can think about this as in bacteria terms, the monocistronic RNA, this is a messenger RNA that codes for one protein, and a polycistronic messenger RNA, it's an mRNA that would encode regions representing more than one gene. So in eukaryotes, we really don't have polycistronic messenger RNAs, but it's a way of beginning to tie in the number of genes with the number of transcripts. Regardless, when we look at the gene number for eukaryotes, we see some interesting kinds of things coming about, in that the bulk of the genes are genes that exhibit some kind of known enzyme activity. The next big guy is different kinds of genes involved in nucleic acid binding. So this is, this is nucleic acid binding, and then this is the genes involved in protein binding. And so we've got a range of different kinds of genes that are doing things that, interestingly, we have a lot of enzyme genes, And then we've got a lot of genes that actually are regulating other genes, what we call transcription factors, or genes that are involved in gene regulation. So if we look at different organisms, it ends up that what we can develop are these ideas of what are known as gene families. That is, gene duplication events occur. And so we can, by looking at sequence homologies, identify gene families. So within an organism, not between organisms, but within, there will be multiple actin genes, as an example, or multiple genes that are sharing very similar sequence, things like receptor genes. There's a whole class of genes that code for proteins known as G protein coupled receptors, or GPCRs, and they all share tremendous amount of sequence homology. They are lumped into a gene family. And so this is another way of estimating the number of types, the number of types of genes. What kind of things are they doing? Another thing that we can look at is orthologous genes. These are genes within different species that are related. So this allows us to establish what is the minimum size of the proteome. What is the minimum number of proteins required by looking at the kinds of proteins that a yeast produces and the kinds of proteins that a plant produces or that a mouse produces. And we can establish what is the basic set. That is, the things you need to carry out glycolysis in the Krebs cycle, the things you need for biosynthesis, the things you need for membrane mechanics and uptake, all these different kinds of receptors, and the very base set of genes that are required. And from this, we come up with some interesting kinds of conclusions. One is that the human genome ends up having fewer genes than was originally expected. And we can explain this away fairly well by the idea that even with these smaller number of gene sets, we can generate a larger, more complex transcriptome. But this is an interesting graph to look at. 1% of the human genome codes for protein. The rest of it is, the bulk of it is repetitive DNA. And then we have introns, and then there is just intragenic DNA sequence between genes, making up another nearly quarter. So this ends up asking some questions of why is this repetitive DNA being maintained? Good answer, or good question. We don't really have great answers for it. One of the things that is starting to show up is that there are sequences that don't look like genes, but they do produce RNAs. They're probably involved in some kind of regulatory systems or control systems. Often you can mutate them and not see an effect, but it's still something that is being looked at still. So the human genome has fewer genes than originally expected, but because of alternative splicing, we can change the size and sequence of proteins, and we've got a much higher number of individual proteins than we do genes. So we have upwards of 1 million different proteins being produced from somewhere around 25,000 genes. So when we start looking at repetitive DNA sequence, this makes up a large portion of the DNA, and this is primarily DNA that is been repeated through the use of what are known as transposons. These are bits of DNA that have the ability to do sort of a copy and paste move within the genome. And as it turns out, there are no active transposons within the human population anymore because it's somewhat unstable. But we have good evidence looking at the genome that these transposons have been present in chromosomes past and replicated themselves around. There are also very large duplications that have occurred, meaning that we have large-scale chromosome breakage and rearrangements to create new chromosomes. And this is actually an interesting evolutionary mechanism of where you've got one gene, And through different kinds of chromosome breaks and DNA replication errors, you replicate that gene. Now you have two copies of it, and you actually are no longer a diploid, you're a quadruploid. You can begin mutating one of those copies, and because you've got the original set, your mutations aren't going to be deleterious. You can begin to develop slightly different receptors that bind different kinds of things. And so by duplicating the genes within a genome, it allows for the increasing in complexity. The Y chromosome shows a number of genes that have been replicated and duplicated within the evolution of mammals, and fine. So a really good question to ask is how many genes are essential? That is, one of the things we know is that you can delete approximately half of the genome, or half of the genes, and not have any detectable effect. What the heck? So when two or more genes are redundant, a mutation in any one of them may not have any detectable effects. All right. So that means to ask how many genes are essential is actually a little bit of a tricky question. We can begin asking through mutagenesis experiments, how many lethal mutations or how many lethal complementation groups can you generate? And this becomes a difficult question because there are genes that may be expressed under very narrow circumstances. There are genes that are expressed only under conditions of extreme stress. And you're growing your organism up under conditions that allow it to grow and thrive. So you put your flies at the perfect temperature, the perfect humidity, they have plenty of food. This is how you can culture them. There may be stress-related genes that you're never going to see. So that's just one way to think about this. We don't really understand why there are genes that are apparently dispensable, but they still remain in the genome. So we've got a whole bunch of genes that actually have no detectable effects. And this is just part of the evolutionary process. So to develop the understanding of how many genes are essential, lots of different experiments have been done, particularly in flies, where you identify lethal mutations. And then it turns out that there are combinations of mutations that by themselves are not lethal, but when combined with others, they actually are. And so this just increases the complexity of the way genomes are working. Okay, so another aspect of the genome is that some messenger RNAs are phenomenally rare. There may be only one or two copies in a cell. And this accounts for a lot of the complexity seen within RNA, and that there are going to be genes that are very scarce, and a bunch of them. And then there are the genes that we see, the housekeeping genes that we see in high numbers in all cells. And so this creates a bias when we're looking at RNA complexity. So we can have this notion of scarcity and abundance. And we've already been talking about these housekeeping genes. A gene is theoretically expressed in all cells. I will just tell you from a functional research perspective, finding a gene that is expressed in all cell types at a reasonably similar level is really hard. When you're doing experiments in which you're trying to quantify the amount of RNA or the amount of a gene that's being expressed in different cell populations or under different conditions. So Dr. Jen does this with some of her tissue culture cells, where you'll take cells and put them under different conditions. You may add a hormone, you may add a toxin, and you'll look to see changes in gene regulation. It's really difficult because you have to have a baseline gene expression level that is expressed throughout and not changing. And then you compare the change up or down of your unknown gene trying to find a baseline gene is always one of the research difficulties, and it's a cause for some concern with the statistics and things. So we've got a whole range of genes that are expressed in very small numbers, and then genes that are expressed in large amounts in very particular cell types. Liver is going to have a lot of certain genes being expressed that muscle cells won't have. And certainly muscle cells will produce a huge amount of muscle myosin that other cells won't be producing. So the genome complexity is still a really critical issue. Now, one of the tools that is still being used is something called a microarray system, in which this is an experimental technology, and it actually has its origins with a dot matrix printer or an inkjet printer, where you can spray onto a glass slide a dot of a DNA sequence. And essentially it's a very small dot that you can only see under a microscope, and use this as a hybridization system. So what you can do is, Affymetrix is a company that does this. You get the Affy chip for humans, and it'll have all 25,000 gene sequences sprayed into a pattern. And you then take RNA from different cell types or under different conditions, whatever. You turn that RNA into a single-strand DNA sequence that you label with a fluor. And you then hybridize it to your Affy chip. And then you look at it essentially under a microscope with a laser fluorescence. And you look to see whether a spot lights up with your gene or during these different conditions. Let's see if we have a picture of this. So this is what a DNA microarray experiment might look like. This is a diagram of 59 invasive breast tumors, and comparing women who breastfed with women who never breastfed. And then the tumor subtypes are denoted by the green, blue, or red. And some interesting experiments can be generated from this. So let's just take a moment here in the last moments and just diagram an experiment you might do. So say you're going to look at what genes are being expressed in a cancer cell. Where'd my eraser go? So this is still a really important technology, that is microarrays. So you've got your Affymetrix DNA chip that has got spots of DNA sprayed onto it. And these are sequences that are from every gene we know in the Human Genome Project. You then develop a set of RNAs. Let's see if we can diagram this. And we'll use red and blue, why not? So maybe we'll take the messenger RNA from a breast cancer tumor, and we'll have messenger RNA from normal breast tissue. Now, what we would do is isolate RNA from these two different sample populations using reverse transcriptase, the enzyme that can take RNA and convert it to DNA, like what we use for cDNAs. And you simply transcribe the gene. You reverse transcribe it in the presence of these fluorescent tags. So not dideoxys, just a fluorescently labeled nucleotide. You then heat it up to make it single-stranded, and you hybridize it to the chip. And you can do the experiment in a couple of different ways. You can hybridize it to two separate chips, or most likely you hybridize it to the same chip. And you might get something to look like this. So you'll have a set of genes that are being expressed in normal breast tissue. And then when you, and the way you visualize these is by looking at these different fluorescent wavelengths. You look at the fluorescent wavelength for the messenger RNA of breast cancer tumors, and you find out that they're showing genes very similarly, but there are a couple of gene sequences that they're not expressing, and then there's going to be a couple guys that are expressing only in the breast cancer genes, or the breast cancer system. Now this is an interesting experiment now. What you've got is a series of genes that are expressed in both populations, and then we have a gene that's not expressed and a gene that has been abnormally turned on. So we want to look at why, or what this gene is doing to control gene expression that is not being turned on. How is it being inhibited? We also want to look at these genes and see what they're doing, because those might be potential targets for some kind of therapeutic. That these are genes that are being produced only in during the conditions of the breast cancer. Another thing that makes this even more complicated is not only do you see this pattern of differential gene expression, you also can see genes that, while they may be expressed in both conditions, the gene may be expressed at a much higher rate or a much lower rate in the different conditions as well. So you can get a lot of information using microarrays. There are other technologies that are coming about in which you can take RNA and sequence it. That can give you some pretty good information as well. But microarrays are a pretty cool trick. Now, another thing that microarrays have been used for, not just for looking at differences in gene expression, but companies like 23andMe, they use microarrays that are specific for single nucleotide polymorphisms. And so you can distinguish between alleles of a specific gene, and that's how you're able to get these different kinds of gene patterns that an individual might have, different kinds of traits. So you might have a gene that codes for curly hair. Well, a gene that is going to be involved in hair texture. And there is going to be a sequence, probably a single nucleotide polymorphism, that is associated with curly hair and one that's associated with straight hair. And you have a little piece of DNA on the chip that's got that single nucleotide polymorphism. And it's not a very big piece of DNA, so the hybridization can distinguish between having that nucleotide or having a different nucleotide. And you carry out very stringent hybridization conditions, and you can actually get SNP profiles using microarrays. So it's another very neat technology. We don't really have the systems to do it, and it is a global way of looking at gene expression and gene expression changes. All right, so that is Chapter 6. There's a little bit of stuff in Chapter 7 we're going to cover in terms of repeat sequences, and after that we're going to get into really some interesting things about gene regulation. All right, that's Dr. G. I'm out of here. I'll be talking to you later this week. Bye.